Okay, great. Okay, why don't we get started? Okay, so it's my great pleasure to introduce Kolade Adebowale, who's a real chemical engineer. So he got his undergrad degree in chemistry in Illinois Institute of Technology, and then a master's at Columbia. And in 2015, he started at Stanford and got his PhD with O.V. Jadhuri. And during his work there and previously, he's done really beautiful work on looking at how various aspects of mechanics affect cell biology, including working on the nuclear piston, and especially on looking at the dissipation in the substrate and how it affects cell biology. And that's one of the things he's going to tell us about today, I guess, in the context of looking at immune function, which is a focus of the new work that since he's moved to a postdoc at the Wyss Institute in Boston. Okay, so let me stop there, and I'm really looking forward to this talk.
Can you see my screen and audio and hear me okay?
Yeah.
Awesome. Let me just hide this. So I would like to start off by, thank you, Paul, for that really great introduction. And I would also like to start by thanking the organizers of this symposium series and the opportunity to present here today. So being here today is actually very personally meaningful to me because I've looked to the mechanobiology community at Penn for a lot of inspiration and continue to do so over the last 6 years. And I'll try to point out some specific connections to this community as I move on with this presentation. And lastly, thank you all for being here today. So today I'm going to be talking about how matrix viscoelasticity regulates cell migration. As Paul mentioned, my name is Kolade Adebowale. I'm currently a new postdoctoral fellow at the Metrology lab at Harvard, but much of the work that I'll talk about today was actually done when I was a PhD student in the Chowdhury lab at Stanford. So here's an outline of the talk today. I'll try to set the stage for 2 vignettes. The first is going to be much longer than the second part, and then I'll summarize, and then I'll take some questions at the very end. Cell migration is an essential biological process that's important for embryogenesis, wound healing, immune cell trafficking, and cancer metastasis. What happens during metastasis is that cancer cells break away from a primary tumor. They migrate through the ECM or extracellular matrix. They enter a blood vessel, exit that blood vessel, and then colonize a secondary site. This process of metastasis is responsible for up to 90% of cancer deaths. But one thing I hope you can appreciate, at least from this schematic, is that cell migration plays an important role during this metastatic cascade, and that importantly, the cell-ECM interaction could be important during cell migration. And so this has led many groups around the world to develop biomaterials that can mimic certain ECM properties and to try to understand how those properties potentially impact cell migration. So some groups have looked at the role of mechanical confinements on cell migration. Others have looked at how the stickiness or the adhesion of a substrate can impact cell migration. Others have looked at how differences in Young's modulus or stiffness can impact cell migration. But the potential impact of viscoelasticity on cell migration is not known. But why is viscoelasticity important? It turns out that normal tissues are viscoelastic, and this was summarized by a recent review article that was published with important contributions from some of the faculty at Penn. And this data is plotting the loss modulus or the viscosity in the material on the y-axis as a function of the storage modulus. And if you look along the diagonal line on this plot, you can see that there are a lot of data points clustered around this diagonal line. And I don't expect you to be able to read all the data, but essentially what this data suggests is that a lot of biological tissues like brain, liver, and lung, and even some of the common proteins we use in the lab, like collagen and Matrigel, are viscoelastic. So they have both properties of liquids, of fluids, and solids. And importantly, these materials are specifically viscoelastic solids, so they're kind of squishy like Jello. But not only is this property of viscoelasticity important in the context of normal tissues, it's also being implicated in diseased tissues as well. So on the graph on the right, you're looking at the loss modulus on the y-axis as a function of the dynamic modulus or stiffness on the x-axis of human tumor samples. What this data suggests are malignant breast tumors are not only stiffer, but they also have a higher loss modulus, which means they have more viscous properties compared to benign breast tissue. And this increase in both stiffness and loss modulus has been found for not only human breast cancer, but also bladder cancer, liver cancer, and pancreatic cancer. One of the consequences of viscoelasticity is that viscoelastic materials exhibit this property, which we call stress relaxation. And we can actually measure this property in the lab by doing a stress relaxation test, where we input a constant strain into a material and then measure the stress response. And so, for example, here's a schematic view of what the results from this experiment looks like, where we're plotting on the y-axis the normalized stress as a function of time on the x-axis. For a purely elastic material, like a rubber band, for example, there is no stress response as a function of time when it's strained. But for viscoelastic materials, like Jello, for example, when these materials are strained, the stress of the material actually decreases as a function of time. So this slide summarizes the main technical portions of the talk today. So in the top row, I'm going to be talking about 2D cell migration, and then I'm going to switch gears and then transition to how immune monocyte cells can migrate in a 3-dimensional context. So now I would like to move on to the first part of the talk today. Lamellipodia have been described as a universal mediator of 2D cell migration. Lamellipodia are these structures that are found at the front of a cell that's migrating. They are characterized by a dendritic actin network, stress fibers, and a very spread cell morphology. And we currently have a textbook view of the critical role that Lamellipodia play, at least during 2D cell migration. And this is summarized by this paragraph on the bottom that essentially says that cells that are not able to make normal Lamellipodia crawl poorly and tend not to metastasize. Over the last several decades, there's been intense focus in the biomaterials community to try to understand how differences in Young's modulus can impact cell migration. What's been found is that if cells are on a compliant substrates, the cells are very rounded and non-motile, shown in this white region. But if those same cells are on a more rigid substrates, the cells can become motile, shown in this light blue region, and that associated with this transformation is a formation of Lamellipodia structures at the front of the cell that's migrating and a very spread morphology. Of note, however, many of these studies have previously utilized substrates that are very rigid or purely elastic. So in this part of the project, we utilize hydrogels with independently tunable stress relaxation properties. These hydrogels consist of 2 main components. The first is basement membrane matrix or matrigel, shown by the green lines, which the cells can actually bind to. And the second component is alginate, shown by the dark blue lines. And this alginate is ionically cross-linked by adding calcium ions. The way that we can modulate the stress relaxation behavior is simply by changing the molecular weight of the alginates. That then allows us to make a range of viscoelastic materials that all have the same initial Young's modulus of about 1.5 kilopascal, or a purely elastic polyacrylamide hydrogel that has an initial Young's modulus of about 1.5 kilopascal. So these materials are very compliant. And to give you a sense of how compliant they are, a gummy bear has a Young's modulus of about 70 kilopascal. So these hydrogels with an initial Young's modulus of 1.5 kilopascal are quite compliant. Importantly, however, the viscoelastic materials have different stress relaxation properties. And we can quantify these differences, for example, by looking at the time that it takes for the peak stress in the material to reduce to 1/2 its initial value. And we call this number T1/2. And this T1 is about 2,000 seconds for what we call a slow stress relaxant hydrogel. That's the data in green. And about 100 seconds for what we call a fast stress relaxant hydrogel. That's the data in blue. So to establish some nomenclature for this part of the talk, whenever you see fast on a graph, that refers to fast stress relaxant hydrogel. In any case, however, we like this material system because it allows us to achieve different substrate stress relaxation while maintaining similar Young's modulus, constant matrix composition, and similar pore size. This is important because later on, if we observe any differences in cell migration, we can attribute those differences to differences in substrate stress relaxation. So now we would like to know what is the potential impact of differences in substrate stress relaxation on 2D cell migration. So we can make these materials and then we can put, for example, human HE1080 cancer cells on top of these materials and then watch how they migrate as shown in this movie. We can then plot the migration trajectories of cells on either a fast relaxing or slow relaxing substrates. So each of these lines you're looking at is the migration trajectory of an individually migrating cell, and they're all plotted on a common axis. And here's a scale bar to give you a sense of how long and how far these tracks actually are. And what I hope you can at least appreciate qualitatively is that the cells are more migratory when they're on a fast relaxing compared to a slow relaxing substrate. Furthermore, we can quantify these differences, for example, by looking at the cell migration speed. And again, what we find quantitatively is that the cells are more migratory on a fast relaxing compared to a slow relaxing substrate. We've obtained similar results with MD8, MB231 human breast cancer cells, as well as MCF10A cells. So these results are not cell line specific. It turns out that not only can the substrate stress relaxation mediates the ability of a cell to migrate, but it appears that it also potentiates the exact kind of migration phenotype. And we found this, for example, by looking at the mean square displacement. So this is a measure of how efficiently a cell is migrating. So a higher mean square displacement, or MSD, indicates a more efficient migration phenotype. And what we find is that this mean square displacement is about 2 orders of magnitude greater when the cells are on a fast relaxing compared to a slow relaxing substrate. From this metric, we can also extract what's called this alpha value. And this alpha value, if it's less than 1, as in the case of the cells on a slow relaxing substrate, it indicates a subdiffusive type of motion. But what we find is that when the same cells are on a fast relaxing substrate, this alpha value is greater than 1, so about 1.2, which indicates a superdiffusive motion. And this is work that we're further exploring with Professor Vivek Shenoy and Dr. Zha Gong, who are both members of the PEN community. So now we would like to know what mediates the differences in observed migration phenotype. And with the exception of one data point on the next slide, for most of the remainder of this portion of the talk, I'll focus only on the results from fast stress relaxant substrates, because that's where we observe the most robust cell migration. One of the things we noticed was that when we looked at the edge of these membranes, we observed these finger-like extensions protruding from the cell membranes when the cells are on a fast relaxant substrate. And these protrusions are filled with actin proteins, and they're highlighted by these yellow arrows in this micrograph. We also observed similar actin protrusions when the cells are on a slow relaxing substrate. In contrast, however, we observe this canonical lamellopodia formation when the same cells are on a more rigid substrate. So this data suggests that lamellopodia are not observed on stress relaxant substrates, but we observe a different kind of actin structure when the cells are on stress relaxant substrates. We then wondered if these protrusions have anything at all to do with cell migration, and it turns out that they actually do. So if you look at this movie on the bottom row, you can see a cell that's migrating from left to right. And as the cell is migrating, it forms these protrusions at the front of the cell. And these protrusions are highlighted by these micrographs over here. It turns out that the number of these protrusions are mechanosensitive, such that there are more of these protrusions when the cell is on a fast relaxing compared to a slow relaxing substrate. These protrusions reminded us of filopodia protrusions. So we stained these protrusions for myosin 10, which is a key marker of filopodia, and we found that these protrusions contain myosin 10 protein. Not only do they contain myosin 10 protein, they also contain additional key filopodia proteins, such as VASP and P130Cas. So together, this data suggests that these protrusions we observe on these stress-relaxing substrates are filopodia protrusions. To further establish the role of filopodia on cell migration, we used a combination of small molecule inhibition and genetic knockdown to perturb the activity of key proteins involved in filopodia formation, and then ask the question, what is the potential impact of those perturbations on cell migration speed by looking at, say, the cell on cell migration by looking at the cell migration speed? What we find is that if we inhibit forming and fashion, the ability of the cells to migrate is significantly diminished. In addition, if we use a more targeted approach by using siRNA to knock down fashion 1 and myosin 10, again, we see that the ability of the cells to migrate is significantly diminished. So together, this data suggests that filopodia protrusions and associated proteins are implicated in cell migration on these fast-relaxing substrates. Perhaps it's not surprising that these cells can form some kind of adhesion with the substrates that they're interacting with. And here we use phosphorylated paxillin as a marker of adhesion. And we specifically focus on the paxillin puncta that are located at the periphery of the cell membrane, which indicate some kind of nascent adhesions. So we find that these adhesions are mechanosensitive on these stress-relaxing substrates. So for example, the number of these nascent adhesions is much greater when the cells are on a fast relaxing compared to a slow relaxing substrates. Interestingly, however, the area of these adhesions is very similar between the substrates, with the adhesions on the slow relaxing substrates displaying a much wider distribution in their area. And then to put this in context of what's commonly done, for example, by looking at what the adhesions look like on the glass substrates, not only do we observe these nascent adhesions, these puncta-like adhesions, but these paxillin puncta also begin to show different morphologies. For example, sometimes they become more elongated or they have different kinds of wedge shapes. In any case, however, there is dramatically greater number of these adhesions on a more rigid substrate compared to the stress-relaxing substrates. And the area of these protrusions is very similar regardless of the substrates. Next, we wondered how these cells can use these adhesions to generate traction forces to potentially support cell migration. And here we did a traction force microscopy type experiments where we embedded fiducial markers inside these materials. And then if I play this movie, you can see that these beads are being displaced in response to cell-generated forces. So these kinds of experiments can allow us to visualize how a cell is interacting with the substrates, for example, by pulling and pushing on the substrates. Furthermore, this traction force microscopy type experiments can allow us to estimate the magnitude of traction forces that the cells are generating on these substrates. And what we find is that the estimates of the maximum traction forces are much greater when the cells are on a fast relaxing compared to a slow relaxing substrates. The next thing we did was we did a few experiments and we found that the cell migration is impaired if we inhibit some of the canonical traction force generation machinery that have been recently studied in the literature. So for example, if we partially inhibit actin polymerization, the cell migration speed is significantly diminished. Similarly, if we inhibit myosin activity, or if we block beta-1 integrins, the ability of the cells to migrate is significantly diminished. So this data suggests that some of the canonical traction force generation machinery are playing a role, at least to some extent, when the cells are migrating on these stress-relaxing substrates. To further obtain additional mechanistic insight, we wanted to implement this motor clutch model of computer simulations in our model. And this is work that we did in collaboration with Professor Vivek Shenoy and Dr. Zha Gong at Penn, and Professor David Odi and Dr. Jay Hu at University of Minnesota. This model consists of three main components. The first is actin polymerization occurs at the cell membrane, and this creates an actin filament and a protrusion. The second component is that there are myosin proteins that bind to these actin filaments and pull this filament toward the cell rear, which generates a retrograde flow of actin. And the third component is that there are these clutch molecules, such as integrins, that the cell can use to sense the mechanics of the material that it's interacting with. The first thing we wanted our collaborators to do was to be able to implement this simulation to reproduce some of the basic migration phenotype that we observed experimentally. And we were really excited when their simulation showed that their mean square displacement, or MSD, is much greater when the cells are on a fast relaxing compared to a slow relaxing substrate. So these trends found in the simulation matched what we observed experimentally. So this gave us a lot of confidence in this model. So now, when I play this movie, we were interested in the dynamics of these filopodia protrusions. And what I hope you can appreciate from this movie is that these protrusions highlighted by the red arrows, they have some kind of dynamic behavior. So they can extend and retract, or even grow longer over time. And so what the simulations predicted is that these protrusions are mechanosensitive, such that the length of these protrusions and the lifetime of these protrusions are both greater when the cells are on a fast relaxing compared to a slow relaxing substrate. We then went into the lab to test these simulation predictions, and we were really excited when we found that experimentally, the filopodia length and the lifetime are both greater when the cells are on a fast relaxing compared to a slow relaxing substrates. So these experimental results matched what the simulations had predicted. So all of the data that I've presented today and some that I've not presented allows us to propose a model. And here we have a cell that's sitting on top of a viscoelastic substrate. And if we sort of focus in on this interface between the cell and the substrate, for example, there are three main components that I'd like to highlight. The first is this actin filament shown by the green rod. And the second component is the substrate shown by the brown rectangle. And the third component is a spring, which is an example would be like integrin, for example, that connects the inside of the cell to the outside world. And the substrate is color-coded specifically because these substrates are stress relaxing. So what that means is if they're pushed on, for example, they have an initial Young's modulus. However, that initial Young's modulus can decrease as a function of time. And that will be represented by a lighter brown color in a second. The other thing that I'd like to highlight is that these springs are being pulled to the left in this schematic due to the retrograde flow of actin. And so those springs begin to experience some kind of load, they come under load, and the springs will be color-coded such that the darker colors indicates a low bond force, and the warmer colors will indicate a high bond force. So as these materials are being pushed upon, for example, in the case of the fast stress relaxing hydrogel, we see that the initial Young's modulus changes from a dark brown color to a much lighter brown color. But at the same time, as I mentioned, these springs are being pulled by this retrograde flow of actin. So they become loaded, and the color changes from blue to red. So they experience a high bond force, another high bond force. But eventually, this mechanical connection is disrupted because these interactions are stochastic in nature. However, this process can repeat itself over and over and over again. In contrast, however, when the cells are on a slow relaxing substrate, this mechanical connection is very prematurely and disrupted early on. And perhaps one way to sort of think about this is that in the fast stress relaxant scenario, it's similar to pulling on a slinky, whereas in the slow stress relaxant scenario, this is similar to pulling on a car strut. So because you're pulling on a car strut, the bonds are loading up very quickly, but they're also failing very quickly. But if you pull on a slinky, for example, it takes time to build load on a slinky, but this time allows you to recruit all the machinery that you require to be able to support cell migration. So to summarize this part of the talk, we have a canonical view that cells are non-motile on a compliant substrate. But in the current work, we demonstrated that even if the cells are non-motile on a compliant substrate, those same cells can become motile if that compliant substrate has sufficient stress relaxation properties, as shown by this light blue region, and that associated with this transformation is the formation of filopodia structures at the front of the migrating cell that actually drive cell migration. And now I would like to move on to the second part of the talk, which is much shorter than the first part of the talk. So here we're interested in how monocyte immune cells can move through three-dimensional matrices. But why are we interested in monocytes? It turns out that monocytes are overrepresented in tumors. So for example, if you look at the white blood cell counts, these monocyte cells are about maybe 7% of the immune cells in the blood. But if you look in a lot of solid tumors, they're disproportionately represented. So because this graph on the right is a little bit complicated, I would like to simplify by only focusing on the proportion of these monocyte cells in several tumors. And what this data suggests is that these monocyte cells can constitute up to about 40% of the immune cells found across a wide variety of solid tumors. So these monocyte cells, for some reason, are overrepresented in the tumor microenvironment. What happens, though, is that these monocyte cells, they get recruited from the blood, as shown by the schematic on the top, where they preferentially accumulate in the tumor microenvironment. But I hope you can appreciate from this schematic that these monocyte cells, they have to physically move through the stromal matrix where they reach the tumor microenvironment that is both stiffer and viscoelastic. And the interaction between the cell and the stromal matrix could be very important during this process. So our goal here is to develop a collagen-based matrix to mimic the stromal matrix that has independently tunable properties. And here we're interested in properties such as the Young's modulus or stiffness, architecture, and loss tangents. The potential impact of stiffness and loss tangents on monocyte 3D cell migration is currently not known. So in this part of the talk, we developed an alginate collagen system with independently tunable properties. The collagen is shown by these light blue lines in this schematic, and that's what the cells can potentially bind to. And the second component is alginate, shown by the dark blue lines, and this alginate is again ionically cross-linked by adding calcium ions. And we can modulate the stiffness or the Young's modulus of this material by adjusting the calcium cross-linker concentration. We can also tune the loss tangents in the material by changing the alginate molecular weights. So I know that this loss tangent is a new term that I introduced. So it's essentially a ratio of the loss modulus to the elastic modulus, and it's a measure of the viscosity in the material. So this strategy that I described in the previous slide allows us to make alginate collagen materials that, for example, have an initial Young's modulus of about 2.5 kilopascal. Importantly, however, these materials have different loss tangent values, as shown by the graph on the bottom left. And for this part of the talk, I'll use a nomenclature where high would refer to a high loss tangent material. So that's the graph in blue on this slide. Because these materials have collagen, they have some kind of fibrillar architecture, and you can begin to visualize what these fibers look like in this matrix from these micrographs. Importantly, we can quantify, for example, the length and the width of these protrusions. And what we find is that the collagen fiber length and width, as well as the Young's modulus, are all very similar. However, the loss tangent is different between these materials. So this data suggests that the matrix loss tangents can be modulated independent of the initial Young's modulus and the fiber length or width. So these results that I've shown are for 2.5 kilopascal matrices, but we've also done similar characterizations for 1 kilopascal matrices, and we have the same conclusions. In any case, however, we can encapsulate human monocyte U937 cells in either a low loss tangent matrix on the top or a high loss tangent matrix on the bottom, and then watch how they migrate. And what I hope you can appreciate, at least qualitatively, is that these cells are more migratory on the bottom. And if you look very closely, you can see that these cells are moving in and out of the plane in a three-dimensional context. But again, we can visualize these differences, for example, by looking at this migration plots that I showed earlier. We can also quantify these differences, again, by, for example, looking at the mean square displacement. And what this data on the top right shows is that if we either increase the stiffness of the material or the loss tangents, either of those independently increase the mean square displacement. And another way to see this is by looking at the cell migration speed. So for example, if we focus on just the low loss tangent material, for example, if we increase the initial Young's modulus, we see an increase in cell migration speed. If we do the same experiment with the high loss tangent material, if we increase the loss tangents, we again see an increase in cell migration speed. And now if we switch gears and look at the impact of differences in loss tangents, if we increase the loss tangents of the material for the 1 kilopascal matrix, again, we see an increase in cell migration speed. And we obtain similar results when we do the same experiments for the 2.5 kilopascal matrices. So again, this data on the right column suggests that if we either increase the stiffness of the material or the loss tangents, we see a corresponding increase in cell migration speed and how efficiently the cell is migrating. To further establish the physiological relevance of our findings, we wanted to encapsulate primary human monocytes in our matrices. And this is work that we did in collaboration with Professor Paul Boyke's lab in immunology at Stanford, working with Dr. Sally Demijan and Medea Pescu. So we encapsulated these primary human cells in our matrices. And what at least I hope you can appreciate from these movies is that the cells are more migratory in the bottom scenario, which is when they're encapsulated in the high loss tangent matrix. And then again, we can visually, qualitatively look at these differences by looking at these three-dimensional migration plots. And we can also quantify these differences where we find that both the mean square displacement and the cell migration speed are both greater when the cell is on a when the cells are encapsulated in a high loss tangent matrix compared to a low loss tangent matrix. It turns out that the morphology of these cells when encapsulated in these matrices are also mechanosensitive. So for example, if we look at the circularity of these cells in these matrices, so the circularity is a measure of how circular the cell is. The higher this metric, the more rounded or circular the cell is. And what we find is that the cells are more circular when they're embedded in a low loss tangent matrix compared to a high loss tangent matrix. Similarly, if we look at the aspect ratio, we find that the cells are more asymmetrical when they're encapsulated in a high loss tangent matrix compared to a low loss tangent matrix. We then wanted to investigate the potential impacts of some of the canonical actomyosin contractility machinery that have been implicated in other kinds of three-dimensional cell migration. So for example, we inhibited ROCK activity, and interestingly, we did not see a significant impact of that perturbation on cell migration. However, if we inhibit myosin light chain kinase, we see that the ability of the cells to migrate is significantly diminished. In addition, if we partially inhibit actin polymerization, we again see that the ability of the cells to migrate is significantly diminished. And then furthermore, if we inhibit the ability of the cells to form this dendritic actin network in these three-dimensional contexts, we find that the ability of the cells to migrate is significantly diminished. So this data on the right suggests that some of the canonical actomyosin traction force generation machinery are playing some role in some of the migration phenotype that we observe. So to summarize what I've talked about today, in the first part of the talk, we utilize a material system that has tunable stress relaxation properties. And what we found is that even though cells are non-motile on a compliant substrate, if those same cells are in a compliant substrate that has sufficient stress relaxation properties, those non-motile cells can transition to a motile phenotype, and that associated with this transition is a formation of filopodia protrusions at the front of the cell that's migrating, and this actually drives cell migration. And in the second vignette, I introduced a set of experiments where we embedded both primary and human monocytes in an alginate collagen system and looked at 3D cell migration. And what we found is that if we independently increase either the stiffness in these matrices or the loss tangent, we see a corresponding increase in cell migration by measuring cell migration speed. And now I'd like to acknowledge my PhD advisor, Professor Obi Chowdhury, all the members of the Chowdhury family, the Boyke Lab, our external collaborators, our funding sources, and also please feel free to add me on Twitter. And just in case if we don't get to all of the questions in the Q&A, please feel free to contact me on email with comments or questions. So my email is kaa@g.harvard.edu. And I'll just say it one more time in case you're listening by phone. My email is kaa@g.harvard.edu. And I look forward to talking with all of you. Thank you.
Oh, that was really fantastic. Okay, let's see. So a couple of questions. Let's start. Phil had a question on the chat. Are you still on? Phil, do you want to unmute yourself? Okay, so if we've lost Phil, okay, can I ask you a question? But
Yeah.
Whether you think that how cell type specific might this be? That if you or yeah, I guess that's the fundamental question. And the second question is, do you think, or have you looked at any kind of fundamental differences between like really malignant cells and normal cells? I know you compared MD231 cells with MCF7s, but do you see a big cell type variation, I guess, in this, enthusiasm for a loss modulus?
Yeah, so I think so far we've only looked at the limited different cell types, to be honest. So just MDM231, MCF10As, and the H1080 cells. Interestingly, when we've also done some preliminary experiments with HL60, which are a neutrophil cell line, as well as U937 cells so both 2D migration. And there, even though I didn't present the data today, we find a similar difference, but it's actually flipped. So the cell migration is enhanced for those immune cells on 2D when they're on a slow relaxing compared to a fast relaxing substrate. So it appears as though this difference in viscosity and dissipation, different cells may react to it differently.
Yeah. So
definitely depending on, say, a cancer cell or maybe an immune cell, they may react differently. But I think the full range of how this actually is mediated is still an unfolding picture.
Great. Thank you. Other questions? Please unmute yourself and speak up.
I have a question and I raised my hand, but I think Shobhan maybe had a question first in the chat.
I'll go second.
Okay, sure. Maybe it'll end up being the same question. Kolade, thanks for the wonderful talk. I have coming from outside of the field, we do some work in rheology, and I guess the question that I have maybe is one that others might have, or maybe you have an answer ready on a backup slide. What is the relevant timescale? So you gave us an interpretation in terms of the loss tangent, for example, but that varies depending on the frequency on which at which you're considering it. And also in the first part of the talk, I thought that what you would tell us is that there is some, and maybe this is coming out of your models, but there is some relevant timescale that's defined biologically, and where that stands relative to the relaxation time or the T1/2 of your gel is really what matters. Is that overly simplistic?
No, I think that's a great point. So some of those relevant timescales, those experiments are a little bit challenging to do, but some of the best estimates that we found from, say, single molecule experiments is that when the cells are interacting with these materials, at least in 2D, they are constantly pushing and pulling on these materials, and the time scale of these interactions have been estimated to be on the order of a few seconds to maybe 1 or 2 minutes. So it's actually a fairly wide range. But I think those are some of the best estimates, that at least that I've read about in the literature. So a few seconds to maybe a minute and a half, 90 seconds. But again, I would also point out still that some of the experiments that I showed with the stress relaxation tests, again, this is so these experiments are looking at the bulk response of the gel. So I think that it's quite possible that the microscopic and nanoscopic response, like the response on the molecular scale, may be a little bit different, like the exact numbers may be a little bit different from the bulk measurements.
Right.
So I think this is an area where some additional microbiological techniques can provide additional insight into what is the cell actually feeling on a molecular scale.
Right, and I get it. There could be, cooperativity and so on that is relevant, and so the fundamental time scale may not be the thing that actually matters, but some other representation of it. But to my second question, or maybe my first question, so the loss tangent that you use to parameterize your response in the second part of your talk, I mean, does it matter at what frequency you consider, you consider it to be defined? It's frequency dependent.
Yeah, so that is a very good point. So we have not looked at that specifically for that material system, but I will show some backup slides for so the first part of the talk, we used a alginate matrigel system. And as you mentioned, we certainly, if you look at the data on in panel D on this slide, so in the bottom right, we definitely see, if we look at the loss tangent as a function of frequency, there is a frequency response. So we have not done these kinds of extensive characterizations for the material in the second part of the talk, but we certainly anticipate that there will be a frequency dependent response in the loss tangent as well. So it does matter what the frequency is.
Thank you.
Yep.
My question, part of my question has already been asked, which is awesome, which is probably what Dave thought was going to happen. I think, and you started to go into this too about not just time scale right now, but also the sort of length scale of relevance, and like what exactly, our tests are measuring when they're sort of bulk rheology tests versus nanoscale tests.
Mm-hmm.
But then I was also thinking with this data here that you have, at least for sort of a large range of those frequencies, the proportions are pretty maintained, like between the different materials. So then I'm just wondering if, when you looked at sort of the modeling and things, whether there was sort of dimensionless parameters that were used, which might also help, because you could imagine that the exact numbers might be different depending on frequencies or depending on length scales. But if they're relatively similar, then you could sort of get away with, you probably end up with similar types of results. So I don't know if you feel like you want to comment on that, or if that's
so I can briefly comment on that. So a lot of the modeling work was done by our collaborators at Penn, but I know that one of the things that I'm not familiar with all the details, but one of the things they looked at is that there is, again, this goes back to the previous question about timescales, for example. So there is a characteristic timescale that the cell is trying to probe its environment and push and pull on this environment. And there's also a characteristic timescale that the material itself is responding to that exogenous force. And what the simulations are suggesting is that if those timescales are very similar, if they overlap to some extent, then we can see this potentiation of this impact of viscosity or dissipation. But if those timescales are very, very different, for example, in the case of the slow relaxing substrates, then we don't see this acceleration or improvement in cell migration speed. So that's what those simulations suggest. And I don't think I have those simulation data in these slide sets, but they've definitely looked at these dimensionalized timescales for the material and the cell itself.
Hello. So I'm very intrigued, but in your first part of the talk, this transition from lamellipodia to filopodia, depending on this viscoelasticity. So, and in this sense, I know that you did inhibitors, but I'm curious whether you also applied CK666 in these conditions, or maybe you stained with markers of lamellipodia, because it is possible that these filopodia just narrow lamellipodia. Structurally, they are different from conventional filopodia. So do you have any information on this point?
Yeah, that's a great point. So we did do some, for the first part of the talk, we did do some inhibitor experiments with CK666. And interestingly, we did not really find any significant impact of that inhibition experiment on cell migration speed. So the second part of your question, we didn't actually stain for any canonical lamellipodia proteins. For example, we didn't do a stain for ARP23, for example. But one of the things that we did was, because our gels are really thick and we have cells on top of the gels, so just to go to your comment, it's very possible that we're just not able to see these lamellipodia because the gels were so thick. So what we did was, later on, we were able to very carefully, through a lot of trial and error, we're able to actually flip these gels without disturbing the cell surface too much. And we can actually image the cell directly from underneath. And even when we did those kinds of high resolution images, we really didn't see any kind of evidence of lamellipodia formation. So that was encouraging. We also tried a series of experiments with lattice sheet microscopy at Stanford. But unfortunately, those experiments, they're a little bit finicky and we weren't able to move too much on that. But a lot of the work that we've done so far, we really don't see any strong impacts of lamellipodia. Yeah.
Could I ask, so the fast and slow and the high and low extremes that you described, like how do those compare to actual tissues? I mean, we all know about the range of, stiffness in tissues, but how do the values that you're talking about, how do they relate to real tissues, and sort of, are there examples of both extremes in real life?
Yeah, so that's a very great question. So I will give a two-part answer. So the first part would be that if we look at this review article that I pointed out, the review article here suggests that this loss tangent is or the loss modulus is about 10 or 20% of the elastic modulus. And so that number, if we compare to the second part of the talk, so here we have I showed the loss tangent values. So this would be the data on the bottom left. Mm-hmm. So those numbers reported for tissues, they're in the range of what we have here. So these are about maybe 8% to 12%. So somewhere in that range. And so that establishes some of the potential relevance to actual biological tissues. Mm-hmm. And I would also mention that we have another collaboration that we did with Professor Natalie Turok also at Stanford.
Okay. Yes.
Yeah. And there we also found, so these are experiments with healthy mouse liver and cancerous liver. Mm-hmm. And again, we find I don't have the data in this slide set, but we find that in this in vivo model, there actually are differences in stress relaxation that are similar to some of the numbers that I showed in the first part of the talk. So in the order of maybe 100 seconds for the cancerous liver and much longer for the healthy liver.
Okay, I actually talked to Natalie about these data last week, so,
so
maybe we should talk sometime.
Yeah, definitely.
That's very helpful to put it in that context for me. Yeah. Thank you.
Let's see. I don't see any hands up. Sorry, I forgot to check earlier. Any other questions? Shaven, I share your pain. These are also experiments that are fantastically interesting to do. We should all be doing them, right? But could I ask you a question on the monocyte? I may have missed this. So I guess this is stuff you're doing with Paul Bolicki. But do you have any evidence that, in addition to the migration, if you put a resting monocyte on a substrate that's either more or less, dissipative, do you change its ability to get activated by cytokines?
So that's a very good question, and the short answer is our preliminary evidence suggests that the answer is yes. So if we put these monocytes, well, I guess this is not putting on, this is more like embedding in a three-dimensional context.
Uh-huh.
So these monocytes are in a more dissipative matrix. We see an upregulation of pro-inflammatory cytokines as well as growth factors as well. So that's really interesting observation. And this is something that we are pursuing further. We also see evidence for markers like INOS, for example, which is a pro-inflammatory marker.
Yeah.
So these are some exciting results that we found recently. I'm just gonna, yeah.
So yeah, we should talk later offline. But there's also these really pretty experiments from Pete Gailey at Rowan, who has been looking at during neural injury, the so-called scar that forms is not stiffer; it's actually more liquidy, more dissipative, and that also drives the inflammatory response of astrocytes. Which is surprising. I would have thought that stiff environment would make things more inflammatory, but it's totally the other way. It's the increase in the fluidity that has some really strong effect.
I just pulled up this slide deck really quickly. So again, these are experiments addressing your question, Paul, some preliminary experiments. So here we're looking at the differential response when these monocyte cells are encapsulated in a more or less dissipative environment. And essentially, there's a lot of texts and acronyms on this slide, but what the data suggests is that there is a slight upregulation in pro-inflammatory cytokines, as well as chemoattractants when they're encapsulated in the more dissipative matrix. And this is only after 24 hours of experiments. So one thing we're trying to do is we're trying to leave these cells for much longer in these kind of matrices and see if there's an even more dramatic response to the dissipation, material dissipation.
Yeah, that's very cool. That's super cool. Let's see. Any other questions? Oh, let's see. Okay. Otherwise, lots of congratulatory comments. Anyway, that was really. Oh, there are. Sorry. Oh yeah. Custom. Yeah.
Yeah. Hi, Kolade.
Hi, Kasav.
Very great work and very nice talk. I just have two open-ended questions. I don't know, like we can connect later on to discuss more, but I just want to put it forward right now. First is like, do you think this property of filopodia generation is something which cancer cells exhibit because they're more plastic? Like, have you tried with non-cancerous cells and seen that they don't change their migratory phenotype when you put them on these viscoelastic gels? And the second question that I have is, do you think there are some mechanosensory ion channels also involved? Possibly not in your work, but when you put cells in viscoelastic materials, like, do you think there are some ion channels which might directly sense the viscoelasticity and modulate the response of the cells?
Yeah, so those are those are really great questions. So for the second part of your question, we haven't looked at ion channels in the first project that I described, but we have looked at ion channels in the second part of the project that I described. And interestingly, we find that if we use small molecules to inhibit some of these ion channels, it appears as though they're playing a role, but we're still kind of ruminating on those data to try to understand exactly what that means and how that fits into the whole story. But it does appear, though, in the second project, that there is a role that ion channels are potentially playing. And then as far as the filopodia goes, as far as I know, a lot of these filopodia protrusions have been described in the context of cancer specifically, and that they are exhibited by cancer cells, and that these can facilitate invasion and cell migration. But there's also some work that's looked at intravital imaging that shows that when the cell is actually trying to enter, when the cancer cell is trying to enter the bloodstream, it's actually doing it with help from what are called tumor-associated macrophages. And there's some preliminary evidence that there are filopodia proteins that are assisting in opening up the endothelial channels to make space for the cancer cell to move through. So it appears as though there's some suggestion that at the minimum, some of the filopodia proteins might be implicated in tumor-associated macrophages, facilitating cancer cell escape into the bloodstream. But a lot of the work that I've seen so far, and I have some supplementary slides on this, is it's all in the context of cancer. Yeah, cancer cells specifically. Yeah.
That's great. Thank you so much.
Mm-hmm.
Shiva, you had another question?
Yeah, I think it's probably pretty quick. So first thing is just to check, because my memory is not good, even in the last hour. We're talking about alginate matrigel gels, right?
Yep.
So do you think that the cells are able to remodel that material at all? Or are you interested in looking at how remodeling might play into sort of a really interesting dynamic here?
That's a really great question. And the answer is yes, we do. So to specifically address that question, so this is a different study that I didn't talk about today. This was published in 2018. This is looking at MDA MB231 cells embedded in a three-dimensional matrix. And what we find, to cut to the chase here, is that these cancer cells, they can extend invadopodia protrusions repeatedly. So extend, for example, here over 40 seconds, and then this protrusion retracts, and then it extends again over a period of time. And that actually this extension retraction cycle is mechanically remodeling this environment for the cell to be able to actually migrate through. So this extension, retraction, and remodeling of the matrix precedes the cell migration in these matrices. Yeah, because these matrices are very nanoporous and the cell is a micron-sized object. But yeah, here we think that the remodeling and mechanical plasticity is quite critical in 3D cell migration.
Super cool. And I guess some of those nanoscale measurements that we were hinting at earlier might actually be able to measure whether you can see, like, what is the scale of mechanical remodeling, right? Like what property is remodeled due to the structure.
That's a great point. So in that study, I don't have the data on here. What we did was, because we have this alginate material, we can actually use fluorescent alginates. And then when we do that, we can look at these channels. We can look at the cells forming these channels. We can, for example, trypsinize these cells, take them out, and we can look at how long these channels are, how wide they are, how long they stay in place after the cell is no longer there. So these kinds of measurements. So these channels from our preliminary estimates are about maybe 50 microns, and they can actually persist and remain open for a long period of time.
Thank you. That's super cool. I'm really excited to dig into this paper.
Thanks for your questions. Yeah.
Great. Thanks to everybody for talks and questions. We're exactly on time. All right, fantastic. Thank you so much. See you later.
Bye, everyone.